Welcome to Scholars Day 2021. I'm Kira Nolder, and today I'll be talking about the intricate relationship between cancer and microorganisms. What is cancer? So cancer is defined by the National Cancer Institute as diseases in which abnormal cells divide without control and can invade nearby tissue. Cancer affects 1 in 3 people in the United States, with an estimated 1,898,160 new cases to be diagnosed in 2021. The American Cancer Society also predicts approximately 608,570 cancer-related deaths in 2021. These numbers break down to about 4 new cases and 1 death every single minute of this year, with the most commonly diagnosed being breast cancer, lung cancer, prostate cancer, and colorectal cancer. Current therapies for cancer include chemo, radiation, targeted hormonal therapy, and surgery. Pathogenic microbes such as bacteria and viruses have been linked to about 20% of cancer cases. They contribute to cancer initiation due to their ability to disrupt natural cellular processes, cause long-term inflammation while evading the immune system, and promote metastasis. Remarkably, some microorganisms are showing promise in their use for cancer treatment through their innate anti-cancer activities, potential specificity for tumor sites, and their ability to be genetically engineered to kill cancer cells. Viruses enter the host cell and directly alter the DNA, causing disruption. Bacteria damage host cell DNA indirectly and lead to cell cycle arrest, which inactivates tumor suppressor genes and activates oncogenes. This triggers the characteristic abnormal cell growth of cancer. DNA damage of the host cell can be caused by one of 4 known genotoxins, which are colibactin, cytolethal distending toxin, Shiga toxin, and bacterial endonucleases. The DNA damage response is responsible for repair of mutations or base pair deletions. The p53 tumor suppressor gene releases proteins that control cell cycle arrest and normal cell death, called apoptosis. Damage to the p53 gene is found in about 50% of cancer patients and results in unregulated cell growth. The mRNA protein complex repairs double-stranded breaks in DNA, and when bacteria prevent it from reaching these breaks, excessive cell growth occurs due to the altered cellular functions. Mismatch repair proteins facilitate the removal of mismatched DNA base pairs. Depletion of these proteins leads to spontaneous mutations in DNA, causing hyperproliferation of abnormal cells. Activation of host signaling pathways by bacteria such as Helicobacter pylori and Chlamydia trachomatis causes mutations of the p53 tumor suppressor gene and overexpression of oncogenes, a staple in cancer initiation. Chronic inflammation due to bacterial infection develops an environment that supports tumor initiation. Free radicals have antimicrobial properties and include reactive oxygen intermediates such as the hydroxyl radical and superoxide, and reactive nitrogen intermediates such as nitric oxide and peroxynitrites that, when released, cause damage to DNA base pairs, leading to mutations and transformation to an abnormal cell type. After infection, cells are depleted and depend on surviving cells and the inflammatory pathways for repopulation. The inflammatory pathway provides the signal for proliferation to surviving cells, which typically includes abnormal cells that were transformed during the microbial infection. Tumor necrosis factor is a cytokine or an inflammatory mediator that induces inflammation that may be responsible for stimulating tumor growth. A study in mice showed that mice with increased tumor necrosis factor developed growth of skin tumors. An extensive partnership between cancer cells and microorganisms allows them to evade host immune defenses. Cancer cells stimulate the activity of immune checkpoint inhibitors that prevent the immune system from attacking the tumor effectively. Bacteria inhibit the antitumor response of natural killer T cells, which are immune cells released in response to infection. Thus, the body is less able to detect and respond to abnormal cell growth. Microbial biofilms are formed when a planktonic bacterium makes contact with a surface of the host tissue and begins to form a monolayer of bacterial cells. The bacterial layer will secrete a sticky extracellular matrix and continue to grow into a three-dimensional shape while each microorganism works together to protect the whole from host immune defenses. Quorum sensing refers to the regulation of gene expression of the biofilm in relation to changes in its microbial population. Bacteria like E. coli produce peptides that act as quorum sensing molecules and promote tumor cell invasion into normal tissues. Biofilm bacteria also secrete toxins that contribute to the epithelial to mesenchymal transition that occurs when cancer cells are transformed from stationary epithelial cells to motile mesenchymal cells. Current cancer therapies such as chemo and radiation are nonspecific and potentially damaging to normal cells. Bacterial therapy has been at the forefront of cancer treatment research because many strains possess natural anti-cancer effects. Non-pathogenic anaerobic bacteria such as Salmonella enterica can invade and colonize solid tumors and lead to decreased tumor growth. Other bacteria such as Salmonella typhimurium damage cancer cell signals and cause increased amounts of inflammatory cytokines like tumor necrosis factor that slow tumor progression. Bacteriocins secreted by bacteria carry a negative charge and selectively bind to positively charged cancer cells while ignoring neutral normal cells. Niacin, a bacteriocin produced by Lactobacillus lactis, has anti-cancer effects on human breast adenocarcinoma, liver hepatocellular carcinoma, and head and neck squamous cell carcinoma. It is non-toxic to normal cells and approved by the World Health Organization for human consumption. Bacterial biofilms can promote and inhibit metastasis. Polysaccharides released by Streptococcus agalactiae into the extracellular matrix of the biofilm prevent cancer cells from adhering to endothelial cells and disrupting the spread. The microenvironment of the tumor is devoid of oxygen and has an acidic pH. Obligate anaerobic bacteria cannot survive in oxygenated regions and actively seek out hypoxic regions, migrating away from oxygen-rich vasculature and normal body cells. When administered systemically, healthy and tumor tissues are invaded. However, bacteria clear from normal tissue within 48 to 96 hours and only accumulate in tumor tissues. Other bacteria, such as Listeria, infect tumor-infiltrating cells that carry them directly to the tumor site. Microbes can be genetically altered for a variety of reasons. To improve safety of the host, pathogenicity of the microbe is decreased by deleting the major virulence genes. Clostridium novyi is converted to its less lethal form with removal of the gene encoding its lethal exotoxin. Generation of an oxotrophic mutant creates forms of the bacteria that will not colonize due to the unavailability of required nutrients. Targeting of the tumor can be improved by attaching antibody fragments that are specific for tumor-associated antigens to the bacterial surface. Modular synthetic adhesins may also be attached to the bacterial surface and contain tumor-specific binding molecules. Bacteria can be equipped with protein plasmids, DNA, and RNA that allow for host expression of therapeutic proteins, ensuring delivery of an adequate therapeutic payload. The genetic manipulation of bacteria to increase its cytotoxicity includes direct genetic manipulation of the bacterium or using the bacterium as a vector for other cytotoxic agents. To increase bacterial toxicity, many strains have been manipulated to express spore-forming bacterial toxin, cytolysin A, which can induce apoptosis in tumor cells. When using bacteria as a carrier, rhenium, a high-energy beta emitter, can be used to formulate radioactive Listeria to reduce metastasis. Oncolytic viruses possess cancer-killing abilities but have inadequate specificity for tumor sites. Bacteria can be manipulated to carry these viruses to the tumor and disseminate them. One study found that E. coli that had been equipped to express the B18R gene increased the oncolytic potential of vesicular stomatitis virus and resulted in decreased tumor growth. Bacteriobots consist of therapeutic bacteria encapsulated by a biodegradable microbead and act as a micro-robot that evades the immune system and travels to the hypoxic tumor to initiate invasion and colonization that results in tumor regression. Bacteria have been indicated for use in cancer therapy for hundreds of years. Beginning in 1813, Vautier claimed that cancer patients exposed to Clostridium perfringens that causes gas gangrene appeared to have significant tumor regression. In 1890, William Coley trialed the inoculation of patients with inoperable tumors with Streptococcus pyogenes, leading to decreased tumor growth. Though bacterial therapy shows great promise, further research is required due to considerable potential for risks, including chance of toxic bacterial sepsis, lack of targeting efficiency, and DNA instability. Genetic engineering is a primary factor in overcoming these obstacles and can lead to the use of bacteriotherapy alongside current cancer therapies. The potential for increased life expectancy and quality of life for patients affected by cancer is a very thrilling possibility that accompanies use of microbes in cancer therapy. Today we've delved into the role of microbes as oncogenic pathogens and the microbial therapies used in cancer treatment. I hope that you found it intriguing and enjoyed discovering just how something so microscopic can have a ginormous global impact.